So for the genomics tool, we have mostly genome and transcriptome data. And again, this is curated from publications or provided by the authors pre-publication. We do a lot of the genomes that are provided pre-pub. We do limit how much access to the genomes there is before publications. So, and that is up to the authors as to what that is. We also curate some expression data. If we get the expression data in the proper format, we can display that. And I'll talk a little bit about that a little later. With the genomic data, the gene, mRNA, and contigs, so contigs are the transcriptome data, those features are searchable within the database. So the annotations, the feature, the scaffold, the names are searchable within the database. You can use these sequences with the BLAST tool. You can view the genomes in JBrowse. We can also view the synteny between genomes in a synteny viewer. The genomes are put through pathway tools. So we have a pathway psych data for all of them. And then if there's transcriptome data, we can use the expression Heatmap tool. I'm going to touch on all of these in this example I'm going to use. So here are the current genomes that are on GDV. A lot of these were provided by authors pre-publication, and we really appreciate it. Some of these are fully available. Some of them, a couple of them were just added, and they're not quite fully available yet. But these are all available. We have transcriptomes. Some of these have some expression data, but all of these are aligned to the genomes and visible in JBrowse as well. All right, so for this example, we are going to see if there's any isocitrate dehydrogenase genes from the TCA cycle in the Draper blueberry genome. So to answer this, we can search the database a couple ways. So I'm going to show these two are kind of the same, different ways of having one starting point. So I'm going to show using BLAST to find homologs. And then also I'm going to show you how to search the annotations that are in the database. And then we are going to narrow down our list using pathway psych, and then view one of the genes of interest in JBrowse, and also look at for synteny with other genomes. So I retrieved a protein sequence for isocitrate dehydrogenase from SwissProt, and I have pulled up the protein BLAST tool. So BLAST is found under Tools, BLAST Plus, and then you choose from BLAST P, BLAST N, T-BLAST X, T-BLAST N. And I'm using a single sequence in this example, so I just pasted it in. But you can upload a file of multiple sequences if you would like to use BLAST that way. I then chose the Draper genome peptides and hit search. And I just want to point out that if you do a BLAST job with multiple sequences, you don't have to sit here and wait for it to finish. We do allow you to bookmark a results page. It's going to be a unique URL for your results, and it's kept on the server for a week after it finishes. Anyway, so here are the BLAST results that you would see on GDV. At the top are the normal download files that you can download like from any BLAST results. And then down here we have a table that has an expandable arrow. And if you click on that, it expands it and shows you the graphic and pairwise alignment that you would normally see in BLAST results. If you click on the green hyperlink over here, it will take you back to the feature details for this mRNA gene. So this is the feature page for the mRNA gene that was matching. And oops. And you can see there's all sorts of links that I'll give a little more examples a bit later. We have homology. This is your match to SwissProt, Interpro scan results, and annotated terms show up. This view will change those items when you click on them. All right, so this is one way to get a list of genes that have been annotated that are isocitrate dehydrogenase. But we can also, instead of doing BLAST, We can also just search the database and the annotations that are stored in the database using the gene and transcript search under the search menu. So here in this example, the search, you can search multiple ways, but for this example, since we're specifically looking at the Draper genome, we selected that genome, and then we put isocitrate dehydrogenase in the search for the keyword. There's also this customized output option on this search where you can specify which columns are shown. So in this case, organism and source are usually default. I deselected those because I know what organism and what genome it is. So I'm just keeping, but I added the BLAST and interpro and go term results. So after I hit search, I get 37 records results, and you can see that it displays all the data stored in the database. For this search, there is an option to download the table or FASTA. So if you were to download the FASTA file, it would be all the sequences for the genes listed on this table if the gene sequences are available in the database. So some of the newest genomes, the sequence data isn't quite available yet because they aren't fully published. For this genome, it is. You can also download the table like before, and you can import it into BLAST and do your sorting and mark it up as you see fit. So from this table that we downloaded, we are going to generate a text file of just the mRNA names. And we're going to use that with Pathway Psyc. So I have my text file of just the mRNA names. And I'm going to go open up Pathway Psyc. So under the Tools menu, open up Pathway Psyc. And so this is the Pathway Psyc homepage. It has all the genomes listed. I only got the first two in the screenshot. Draper is the one we're going to select. And then once we open the Draper genome, we're going to go to the Metabolism menu and choose Cellular Overview. And this is it then displays a graphic of the whole cell and all the pathways that are available in this genome. And so we see that the TCA cycle is here. Just to point out for Pathway Psyc, if you have omics data, so transcriptomics or metabolomics data that use the same names as are available in this database, you can overlay that data and it will do heat map coloring for the pathways that have expression data or metabolomics data. I'm not going to demo that today. Today, I'm going to demo highlighting genes. So you expand that menu and there's an option to upload a file. So I uploaded my file, and this is a little video because it was easier than screenshots, and it shows my list here of my file I uploaded and says they're going to be color coded in red. So I can see that my file is there, and I can see there's some of my genes in that file are here, and then some of them are over here in the TCA cycle, and you'll notice it gives me a list of the genes that are involved with the TCA cycle. So I've narrowed down my list of 37. So let's look at one of the genes from this list that are involved with the TCA cycle a little closer. So to do that, I'm going to take the gene name I want and paste it into the gene and transcript search under gene and transcript name. And then that's going to return one result because everything's uniquely named in the database. And there's two hyperlinks. So there's two different directions we can go from this page. The first is we can go to location and view the gene in JBrowse. So we're going to click on that one first, and we'll come back and look at this other link here in a second. So here's the gene in JBrowse. That's the top track. And I also have two transcriptomes from V. virgatum aligned to this genome in JBrowse. And one of them has a transcript that looks like it's in the same location, about the same size. So if we click on that feature in JBrowse, it opens up a details page, and there's a purple hyperlink in JBrowse where you can click, and this will take you back to the GDV database about the information about this specific scaffold or contig. So if we click on that, we then get the feature page for this transcriptome transcript, and we will see if we and this transcriptome actually has expression data stored in the database. So if you click on the expression, it shows you the expression data. This is from a study of leaves, berries, and buds. It shows the data here. You can do change the view here. But this expression data can also be viewed under the tools menu using the expression Heatmap tool. And so this is an example of the interface. You either filter your data or you just paste in the mRNA names you want. So we could have also pasted in our list of mRNA names for isocitrate dehydrogenase, but I don't know if all those genes have expression data. Anyway, so you paste in the thing here, you go display expression heat map, and it will actually generate a heat map figure for you that you can download and save and use in presentations. And all the treatments and mRNA names are clickable for more details. So I just wanted to kind of give you a little expression heat map preview. We don't have a ton of expression data. I'd like to get more, but we do have some of it in the database. All right, so back to our gene of interest. That was isocitrate dehydrogenase and the gene and transcript search. We did this link previously. We're now going to do this link over here. And that's going to take us to the details page for that mRNA feature. And you will see that there's a lot of, we have the interpro scan results, we have homology with Swissprot, we got annotated terms. We also have this relationship section, which I'm going to click on. And this is, it's actually longer. It's a longer list than just this, what I'm showing. But I wanted to point out the synthetic regions region here. And you have to kind of learn what the abbreviations are, but VCD to VCD is Draper to Draper. So this is a synthetic block within the same genome. Down here, we have VCD to VMI, that's Draper to Vaccinium microcarpum. And then we have VCD to VMS, so that's Vaccinium, Draper genome to Vaccinium macrocarpon, Stevens. So these are synthetic blocks from an analysis we've done. And so and this gene is in these synthetic blocks. So let's look at the synthetic block against cranberry. And when you click on that, it opens up the SNTNI viewer and gives you a side-by-side comparison of that synthetic block and shows you the corresponding genes. It also gives you a list of, of the genes that are synthetic to each other. So gene A is our Draper genome, gene B is cranberry. And again, any green hyperlink is clickable and gives you more details. So going back to our gene of interest, which is this one right here, it's at the very bottom of the list. It corresponds to this gene in cranberry. So we can click on this. And this is the mRNA feature page for that cranberry gene. And sure enough, if we look at the annotated terms, it's also isocitrate dehydrogenase. So we found our ortholog in cranberry. All right, so that was kind of a convoluted, complex example of how to use the genome tools. But we did prove that there is a isocitrate dehydrogenase gene. There's several of them in Draper, and that we do have homologs in other genomes. All right, so I guess I'll take questions if there are any.